right, we are going to wrap up our exploration of circulatory organization by examining the circulatory systems of the invertebrates. And we'll start out looking at the invertebrates that have closed circulatory systems, and then we'll take a look at invertebrates that have open circulatory systems. And there's also going to be a couple of surprises on our way as we explore these circulatory systems. So we'll start out exploring circulatory systems in the octopi and the squid, so our cephalopods. These groups have closed circulatory systems, and they help us understand that there's no rule that you have to have all of your pumping action in a single location. So the octopi, for example, have a grand total of 3 different hearts. And so they have 2 hearts that feed into their gills. So those are pictured on the left and on the right-hand side of this diagram. Or in our circuitry diagram, they're over here, just ahead of the gills. Okay, so hearts that help pump blood through the gills. That blood then returns, and they have a systemic heart, which will then pump that oxygenated blood throughout the rest of their tissues. And cephalopod circulatory function is generally pretty similar to fish circulatory function. So this is a high pressure system, and remember, think back to that oxygen equilibrium curve for octopi, right? So they're using their full oxygen equilibrium curve even when they're at rest, suggesting they need really good oxygen delivery to their tissues. So to get that good oxygen delivery, right, if you want a good blood flow rate, then what you need to do is make adjustments across this equation for flow rate, right, thinking about resistance and pressure differentials. Well, one way you can bump up your flow rate is by having a very high pressure system. This is also a relatively high resistance circuit, just the way that their circulatory architecture is laid out. So they need that high pressure system to help maintain high flow rates in the context of a high resistance circuit. Now let's kind of change gears a little bit and take a look at open circulatory systems. So everything we've talked about so far has dealt with closed circulatory systems. Blood stays in vessels, it never leaves those vessels. In open circulatory systems, turns out that doesn't have to be the rule. You can also have blood that can move directly through your tissues. To give you an example of an organism with an open circulatory system, I've got a decapod crustacean pictured here, like a lobster. And so in these guys, they still have a functional circulatory system. They just don't have blood in veins and arteries, okay? So just to show you that these circulatory systems still move blood or lymph through their bodies, here's a photo of circulatory structure that's had dye injected into the heart, and you can see that dye is diffusing. It's moving through the vessel pictured here, and it's diffusing into the tissues. And in spite of the fact that blood doesn't stay in vessels, this blood movement can still be strongly directional, and that can be managed by a combination of valves and how muscles constrict. So here I've got a circuitry diagram for a lobster's circulatory system. We can start at the heart. The heart will squeeze, it will send blood to arteries. From there, the blood will filter through the systemic tissues. It will return to the interbranchial sinus, so it's not just going every which way, but there is actually a return path. It will pass through the gills, it will become oxygenated, and then will travel back through the heart before again filtering through the tissues again, okay? And realize this is kind of backwards from what we saw from the fish, right? So in the fish, we had deoxygenated blood reaching the heart and then getting oxygenated, then reaching the tissues. Here we've got oxygenated blood traveling to the heart and then traveling back out to all of the tissues. And this open circulatory arrangement, right, in contrast to what I just showed you for the cephalopods, is a very low pressure system. let's think about it this way. If you puncture a lobster, it's not going to instantly bleed to death. So the circulatory system is relatively low pressure. But just because the circulatory system is low pressure does not necessarily mean circulation is slow. And that's because this circulatory structure also offers relatively low resistance. If we come back up to this equation for flow rates, right, you could get rapid flow by very, very high pressure in the context of high resistance, but you could also get rapid flow in the context of a low resistance circuit, okay? And so if we take a look at the blood flow rates through the systemic circuit for a range of different types of animals, what we will see is that even though these circuits have much lower pressure, the overall blood flow rates that these organisms can sustain are much higher than the blood flow rates that fish can sustain. So on this picture, I've got a number of different parameters. You could spend a lot of time scrutinizing them all in detail. Let's just focus here on the rate of blood flow through the systemic circuit. I've got spiny lobsters, rock crabs, right, or decapod crustaceans. Then I've got a flounder and rainbow trout. And notice how much higher blood flow rates are in the lobsters and crabs relative to these fish, right? And that's, again, in spite of the fact that they have, relatively low pressure systems, okay? So we're going to wrap up now talking about one more group of invertebrates, the tracheate arthropods, right? So these are the arthropods that breathe using a system of tubes that run through all of their tissues. So not bothering with getting gases into a liquid, then transporting that liquid to the tissues, right? So in the tracheate arthropods, functionally, the ventilation system is decoupled from the arthropod circulatory system. And kind of from an evolutionary standpoint, this lets us ask a really interesting question, which is a question of, to what extent is an animal's circulatory structure and its function driven by the demands of gas exchange? And this question will make sense in a second, okay? So to what extent is circulatory structure and function driven by demands of gas exchange? If it is very strongly driven by gas exchange, then we need to also know something about oxygen demands in the tracheate arthropods, okay? And to help think about this, you need to know that some flying insects, like the honeybee that's pictured here, are among the most metabolically active animals in general. So pound for pound, these guys are burning a lot of oxygen as they fly. And they contrast with the crustaceans I just showed you that are generally pretty sluggish in terms of their activity levels. Okay, so what that all means is the tracheate arthropods have very high oxygen demand. So now, if circulatory structure and function are largely driven by the demands of gas exchange, here's an organism that has decoupled breathing, gas exchange, from its circulatory system. And remember that generating pressure in a circulatory system is energetically very expensive, right? It takes a lot of energy to squeeze, a heart, squeeze other vessels. And these guys still do have structures that facilitate movement of fluid through their tissues. All right. So what we find when we examine circulatory function in the tracheate arthropods is that these guys have extremely sluggish circulatory systems. Okay? So if you decouple your gas exchange requirements from your circulatory system, you're keeping in mind your circulatory system still does a couple of other things, right? It's involved in nutrient exchange, waste removal, there are immune functions, right? But if you decouple take gas exchange out of the equation, circulatory function slows down dramatically. So collectively, the big conclusion here is that a lot of what we've seen for circulatory structure and function is a direct product of the demands of gas exchange across animals. And I just think that is just an amazing natural experiment that has let us draw that conclusion. Okay, that brings us through our exploration of circulatory alternatives across the fish, amphibians, and non-bird reptiles and invertebrates. So the next topic that we'll be diving into is the physiology of diving in mammals. The great thing about our next topic is that this is going to help us synthesize the ideas we've been exploring all the way through this point of the semester. We're going to be thinking about gas exchange and circulatory function in a particularly amazing group of animals, the diving mammals.